Coral reefs are one of the ocean's most diverse and ecologically significant habitats. However, given limited management resources, ecologists and conservationists want to prioritize their efforts in specific spatial regions of concentrated biological activity known as hotspots, like this pillar coral on a reef in the U.S. Virgin Islands. The complex 3D structure of the pillar coral creates a haven for different fish species to find food or shelter. However, seeking out and precisely mapping the extent and magnitude of hotspots like these remains a difficult task. Visually, hotspots appear as areas where there are large numbers of marine organisms. Near the pillar coral, you can see many different species, such as sergeant majors, French grunts, and a barracuda. Acoustically, hotspots are the spots with the most active soundscape. In this example, you can hear low-frequency fish calls and high-frequency snapping shrimp crackling. We can encode this understanding of spatial hotspot heterogeneity in a generative model of species-dependent visual and acoustic observations. The generative model then informs the design of 4 different hotspot discovery behaviors that use the different sensing modalities in open-loop mapping and closed-loop seeking modes. Our AUV, CUREE, has forward and downward-facing cameras as well as a planar hydrophone array, allowing it to simultaneously see and hear the reef. Onboard computation with an NVIDIA Jetson GPU enables CUREE to execute each behavior fully autonomously without the need to be tethered to the surface. We demonstrate each of these 4 behaviors in a case study of Joel Shoal Reef in the U.S. Virgin Islands, targeting a potential hotspot location on the northeast side of the reef near the skeleton of a large pillar coral. The first behavior we will demonstrate is a visual fish census. Using a pre-trained YOLO neural network, CUREE detects and counts fish in the downward-facing video feed. The more fish it sees, the higher the likelihood that it's found a hotspot. As CUREE conducts the census survey, we build a map of where and how many fish CUREE sees. As you can see, the edge of the reef near the pillar coral is the most active area, with over 24 times greater fish density than in the surrounding reef. While the census gives us a precise map, building it is slow due to the limited visual range and field of view of the cameras. Our second behavior, acoustic hotspot mapping, addresses this problem by using longer-range acoustic sensing. The challenge with building a similar acoustic map of the soundscape is that we can't localize each sound with the same precision. The hydrophone array is capable of determining the bearing to a sound source by comparing the sound wave's arrival times at different hydrophones, but without knowing how loud a source signal is, range information is lost. To build our acoustic map, we fuse multiple bearing observations taken from multiple observation locations, triangulating the location of each acoustic source. In this heat map, you can see that once again, the area around the pillar coral is the most active hotspot region, with nearly 2 times the density of fish calls as the remaining reef. However, this acoustic mapping and the visual census assume we already know the rough region where a hotspot might lie and are less effective for discovering new hotspots. Closed-loop seeking behaviors allow us to find hotspots in completely unexplored regions. Our third behavior, acoustic homing, finds these hotspots by moving towards areas with active soundscapes. Since CUREE's thrusters mask the natural reef sounds as it moves, we use a listen-then-move strategy. While CUREE is stopped, it listens and computes the bearing of the loudest sound source within the biological frequency bands, 50 Hz to 2 kHz for fish, and 4 kHz to 20 kHz for shrimp. CUREE then moves a fixed distance toward its detected source, and then goes back to listening, starting the process over again. Here are the trajectories showing the emergent behavior as CUREE homes in on an acoustic hotspot. Both homing trajectories first converge on the area with the pillar coral before continuing towards the center of the reef, suggesting that these locations are biological hotspots. Our fourth and final behavior is visual tracking of sentinel species. Since underwater vision is range limited, we can't do the same type of long-range homing that we can do acoustically. So instead, we leverage the experience of the reef's inhabitants by tracking them as they move around the reef. Places they revisit over and over again are more likely to be hotspots. Using the Keep Track Fast algorithm, a semi-supervised object tracker, a human operator can select a target organism, and then CUREE uses visual servoing to keep the target centered in the forward-facing camera frame. By examining the trajectory of the sentinel organism, in this case a barracuda found near the pillar coral, we can see that it first moves away from the reef before ultimately returning back to the pillar coral, once again highlighting it as a hotspot location. Across all 4 behaviors, the area around the pillar coral was consistently indicated as a hotspot location. Using mapping behaviors, we were able to quantify the magnitude and extent of this hotspot using precise visual sensing and long-range acoustics. Our seeking behaviors were able to locate the hotspot with no prior information using acoustic homing and by tracking sentinel species. We hope that you have enjoyed this presentation of our paper, Autonomous Seeking and Mapping Coral Reef Biodiversity Hotspots with a Multimodal AUV. Thank you for watching.